students. Today we will be discussing about one of the important results of photosynthesis. So what is the net production, or what is the importance of photosynthesis? Photosynthesis, it helps in the synthesis of energy-rich molecules such as ATP and NADPH. So how the ATP is produced? ATP is produced through the photophosphorylation, which occurs due to the light-induced electron flow. So today we will be discussing about the mechanism of photophosphorylation and the organelle where it takes place. So photophosphorylation, let's come to the term. The term photophosphorylation means synthesis of ATP in presence of light. Now let's come to the discoverers who discovered this process. So it was first described by Samuel Ruben in the year of 1943, which was later supported by Emerson through their experiments. However, when they were working with the intact chlorella cells, they found that the sole function of the light energy is to produce energy-rich phosphate bonds. However, this theory was opposed by Rabinovich in the year of 1945, and he said that the process of photosynthesis is not thermodynamically favorable. He therefore nullified the theory of photophosphorylation and said that it is impossible, as it is converting light quanta, which is having an energy of 43 kilocals per Einstein, into phosphate quanta of only 10 kilocals per mole. He suggested that such kind of a process would not be biologically sound in photosynthetic organism. However, with the further experiments going on, it was proved in the year of 1954 when scientists were working with intact chloroplast isolated from spinach, which could fix carbon dioxide. So it led to the discovery that it can convert light energy into chemical energy and trap it in the pyrophosphate bond of ATP. Thus, indeed, photophosphorylation is occurring in green plants. So photophosphorylation was discovered with the isolated intact chloroplast, to which no cofactors were added to increase the rate of ATP formation. Now, without the addition of cofactors, the integrity of the chloroplast structure was essential for ATP formation. Chloroplast fragments which showed strong Hel activity but showed very weak rates of photophosphorylation activity. Thus, the intact chloroplast structure was required for photophosphorylation. So how did the photophosphorylation got initiated? It got initiated with the help of light. Light induces an electron flow, which we know now as the Z scheme, and this electron flow then produces a proton movement. This proton movement is responsible for the production of ATP. Hence, there is a light energy had been used in photophosphorylation before ATP synthesis and not related to oxygen evolution. So the most probable mechanism behind the electron flow is the light-induced electron flow. Now, if we look into this diagram, so it shows that how the reaction centers, ATP complex, are arranged on the thylakoid membrane. Now, this thylakoid membrane is impermeable to proton. Hence, when there is an electron flow, there is a movement of protons across the membrane. So this helps in the formation of ATP, that is through the process of photophosphorylation. Now, such a kind of a process also occurs in another organelle, which is mitochondria. Mitochondria, it also shows oxidative phosphorylation. Hence, both chloroplast and mitochondria, they are inducing an electron flow. Through this electron flow, there is a net movement of protons, and this net movement of protons is causing the ATP formation. So if we compare the both processes, so they share some kind of similar features. They are the reaction centers, electron carriers, ATP forming enzymes. These are all located on a membrane, and this membrane is impermeable to protons. The membrane must be intact to support phosphorylation. Photophosphorylation occurring in chloroplast can be uncoupled from electron flow by reagents that promote the leakiness of the membrane to the protons. Again, the electron flow is coupled with photophosphorylation can be proved if there are certain reagents or certain chemicals are used which stops the ATP synthesis by binding to the ATP synthase complex. Thus, the electron flow will also be stopped. The fourth point being the morphology of the ATP synthase, both in case of chloroplast and mitochondria, are the same kind of structures and they are present, and the proton movement takes place from the positive towards the negative side. So, thus we see that the phosphorylation occurring in chloroplast and mitochondria both shares some kind of similar features. Now, the electron transferring molecules which are present in the chain of carriers connecting PS2 and PS1 are oriented asymmetrically on the membrane. So, hence when there is a light induced electron flow, it results in the net movement of protons from the stromal to the luminal side of the membrane. And when there is an ATP synthesis, the protons moves in the opposite direction, that is from the lumen to the stromal side. And during this movement, the ATP is synthesized from ADP, that is adenosine diphosphate, and PI, inorganic phosphate, with the help of this ATP synthase enzyme. Now, if you come, such kind of asymmetric movement of protons also takes place in other organelles as mitochondria, present in the eukaryotic system and responsible for oxidative phosphorylation, as well as in bacterium such as in E. coli. So, this kind of asymmetric movement of protons is the main key factor behind the ATP synthesis. The mechanism of electron transfer occurring during the light reactions has also been proved with the use of herbicides. So, there are chemical compounds known as herbicides which stops the electron flow along the Z scheme. The herbicides are the different group of chemical compounds which vary in their mode of action. Some herbicides, they act on the carotenoid biosynthesis, or they inhibit the amino acid biosynthetic pathway, or they inhibit the lipid biosynthetic pathway. Herbicides such as DCMU or paraquat, commonly known as the methyl valogen, they inhibit the Z scheme at various points. DCMU is known to inhibit the plastoquinone binding site at the photosystem 2. So, it stops the electron flow at this particular point. Similarly, paraquat accepts electron from photosystem 1 and it gives the electron to the oxygen producing reactive oxygen species. Hence, it proves that there indeed is an electron flow occurring during the photosynthesis, which is commonly referred to as Z scheme. Now, following this, then came the big famous theory behind the ATP synthesis. So, this theory came to be known as the Chemiosmotic Theory, which was put forward by Peter Mitchell. So, Peter Mitchell, he said photophosphorylation works via chemiosmotic mechanism proposed in 1960s by Peter Mitchell. Peter Mitchell, he said the chemiosmosis is the unifying force behind the membrane related processes occurring in different forms of life. That is, according to this hypothesis, the electrochemical energy is the reason behind ATP formation. So, if you look into the chemiosmosis mechanism, so chemiosmosis mechanism occurs in two different organelles of eukaryotes, that is mitochondria and chloroplast. Both mitochondria and chloroplast, they generate ATP via a protonomotive force resulting from an electron flow. The protonomotive force results in an electrochemical imbalance or gradients across the membrane. Both of them utilizes an electron transport chain, including a cytochrome mediated electron transport, and this cytochrome in the process of electron transport pumps H+ ion across a membrane. Both of them use a similar kind of ATPase or ATP synthase complex. So, chemiosmosis is occurring behind any membrane related processes which are responsible for ATP generation. Now, let us come to the protonomotive force. So, what is a protonomotive force? Protonomotive force, if you look into it, it basically consists of an electrochemical gradient. So, the electrochemical gradient which is built up due to the diffusion of or pumping of H plus ions, that is the protons, across the membrane. So, protonomotive force consists of two kinds of forces, the chemical potential gradient and a transmembrane electrical gradient. So, these two makes up the electrochemical gradient. These two components of the protonomotive force travels from one side of the membrane to the other side. The basic principle of chemiosmosis is that ion concentration and electrical potential differences across the membrane are a source of free energy that can be utilized by the cell. The asymmetric nature of the photosynthetic membrane and the fact that the proton flows from one side of the membrane to the other side accompanies electron flow as studied by different experiments. So, this can also be studied or verified or confirmed with the help of uncouplers. So, this is about the protonomotive force. Now, let us come to the equation describing the protonomotive force. That is, delta P is equal to delta E minus 59 delta pH. So, what is delta P? The delta P stands for the protonomotive force, delta E is the transmembrane electrical potential, and the pH, that is the delta pH, is the pH difference across the membrane. The constant of proportionality at 25 degrees centigrade is 59 millivolts per pH unit. So, a transmembrane pH difference of 1 pH unit is equal to a membrane potential of 59 millivolts. So, this is how the protonomotive force is built up. The stoichiometry of protons translocated per ATP synthesized has been found to be 4 H plus ion per molecule of ATP synthesized. That is, a diffusion of or a movement of 4 H plus ion across the membrane produces 1 molecule of ATP. Now, let us come to an important experiment where it was proved that the pH gradient drives ATP synthesis. This was proved by Andrew Jagendorf, and this helped Peter Michel to develop the chemiosmotic theory. What he did? He isolated intact chloroplast from a particular plant material. Then he incubated this chloroplast into a buffered medium which had a particular pH of 4. So, hence now what happened? Due to the presence of pH 4, so this protons diffuses into the chloroplast lumen, the lumen of the thylakoids, and it reduces the pH of the thylakoids to pH 4. After this, the chloroplast suspension was incubated in dark, and then a mixture of ADP and PI was added to the chloroplast suspension. Next, the chloroplast suspension was then transferred to a buffer which was equilibrated at pH 8. Hence, there is a change from pH 4 to pH 8. Now, what happened? So, this chloroplast culture was incubated in the dark. When it was incubated in the dark and there is a pH difference, that is pH 4 to pH, the outside is pH 8, hence it produced ATP in absence of light, and this is how photophosphorylation works within the plants. So, light is only inducing an electron flow. This electron flow is inducing the pumping of protons, and this pumping of protons is producing an electrochemical gradient known as protonomotive force, and this protonomotive force is producing ATP with the help of ATP synthase complex. This experiment helped Peter Michel to understand that the reason behind ATP synthesis is not light, but it is basically the pH gradient, which induced the ATP synthesis. Such as the similar kind of a thing is also occurring in vivo within the plant cells. If you now let's move to the structure of ATP synthase in chloroplast resembles very similar to the ATP synthase found in mitochondria. It is known as the ATP synthase or CF0-CF1 complex present in chloroplast. Let us see it denotes its location in chloroplast, whereas the O stands for oligomycin sensitive. That is, when it is treated with oligomycin, it inhibits the ATP synthase complex. Let's come to the parts of ATP synthase. The ATP synthase consists of CF0, CF1. CF0 is present in the membrane. It is a mixture of integral membrane proteins or transmembrane proteins, and CF1 is produced or appears as a knob which protrudes towards the stroma portion of the chloroplast. So, the similar kind of an ATP synthase also is present in mitochondria, which is known as F0-F1 complex or ATP synthase. So, in general, the movement of protons takes place from the positive to the negative side in the case of both chloroplast and mitochondria. Now, let us come to the individual structural units of CF0 and CF1. Now, let us first start with CF0. As I said, CF0 is made from integral membrane proteins. If you look into the structure of CF0, it consists of the subunits A, B, C. These are single copy of A, 2 units of B, and the C can be present in 10 to 12 units. So, it is A, B2, C 10 to 12. Now, similarly, the CF1 is made from the membrane units. The composition of CF1 is, it is made from 9 units, that is alpha, beta. So, there is trimeric alpha, beta subunits along with gamma, delta and epsilon. So, this is how the CF0 and CF1 complex are associated with each other. Now, first again come back, or let us come back to CF0. The CF0, so A and B proteins are helping in fixing the CF0, CF1 complex on the membrane, whereas the subunit C, which is arranged perpendicular to the plane of the thylakoid membrane, the subunit C is present in 10 to 12 copies which are arranged in 2 concentric circles. The 2 concentric circles, which has also been studied in different organisms, especially in yeast, it has been found that the C subunits are present in 2 circles. The inner circle, which is made from the amino terminal helices, whereas the outer circle is made from the carboxy terminal helices. The gamma and epsilon, or the delta and epsilon, they protrude out from the base of CF1 and sit on the circle of the C subunits. So, the proton pore is present within the CF0 subunits. Proton pore refers to the pore through which the protons are translocated from the lumen to the stroma. Now, coming back to CF1, the CF1, as I had already told you, it consists of trimeric alpha beta subunits arranged very similar to the section of an orange. Now, this is the beta subunit each beta subunit has a catalytic site. The catalytic site of the beta subunit was studied, or the crystallographic determination of the structure of F1 was studied by John E. Walker. The CF1 is a flattened sphere measuring 8 nanometer high and 10 nanometer across. So, it is arranged like the sections of orange. The polypeptides which connect CF1 with CF0 are asymmetrically arranged, with one domain of the gamma subunit making up the central domain or the central shaft, and another domain of the gamma subunit is in contact with one of the beta subunits. So, the domain of gamma subunit which is in contact with the beta subunit is referred to as the beta-MT conformation. Now, all the 3 beta have the similar kind of amino acid composition, but they differ in their conformations. So, it is due to this conformations that the 3 beta they bind with different structures of ATP. Now, let us come to the mechanism by which the ATP is synthesized by this CF0, CF1. It is known as a rotational catalysis proposed by Paul Boyer. So, he proposed a rotational catalysis mechanism in which the 3 active sites of F1 takes turn in synthesizing or catalyzing ATP synthesis by complex 5. The conformational changes central to the mechanism are driven by the passage of protons through the F0 portion of ATP synthase. So, the 3 beta subunits interact in such a way that when one is assuming the beta empty conformation, then the neighboring beta subunit is in the ADP form and the third beta subunit is in the ATP form. Thus, one complete rotation of the gamma subunit, or when there is a passage of 4 protons across the CF0, then it is moving the gamma subunit, and the gamma subunit which is in contact with each beta subunit to cycle through all the 3 conformations. That is, each beta subunit passes through the 3 different conformations, its binding of ADP and BI. The next it moves towards the tightly formation of ATP, and in the third conformation it changes to the empty where the ATP leaves the beta catalytic subunit. So, thus each by 120 degree each beta subunit passes through 3 different conformations. With each rotation, thereby 3 ATP are synthesized and released from the enzyme surface. So, 4 protons is catalyzing 1 ATP synthesis and 120 degree is synthesizing 1 ATP. So, a total of 360 degree is synthesizing 3 ATP. So, if you compare the bioenergetics of cyclic and non-cyclic photophosphorylation, it is found that from bioenergetics point of view, cyclic photophosphorylation is more profitable than non-cyclic photophosphorylation. Hence, the cyclic photophosphorylation is of more energetically profitable for the plants using less energy and producing more ATP in comparison to non-cyclic photophosphorylation. Now, let us conclude then. So, we saw that how light-induced electron flow along the Z scheme helps in the movement of protons across the membrane. The movement of protons takes place at 2 different sites in the Z scheme. One during the photosystem 2 or at the photosystem 2 where there is photolysis of water, and another where there is cytochrome B6F complex. Now, the net movement of protons takes place from stroma to the thylakoid lumen. As a result of accumulation of protons more inside the thylakoid lumens, it drives the pH more acidic, or this side of the membrane becomes more positive, whereas the stroma becomes more negative. So, this accumulation and asymmetric distribution of protons produces an electrical and a chemical gradient. Together they are known as the electrochemical gradient. Since the electrochemical gradient is made from protons, it is referred to as protonomotive force. This protonomotive force then flows from lumen towards stroma with the help of ATP synthase complex. In the process, when it is flowing from stroma to the lumen, then it is producing ATP with the help from ADP and BI. The complex is referred to as the CF0, CF1 complex, very much similar to the mitochondrial one. So, this is how the light induces the synthesis of ATP, referred to as the process of photophosphorylation. Photophosphorylation are of 2 main types: non-cyclic, cyclic. It is a cyclic photophosphorylation which from the bioenergetic point of view is more profitable for the plants, which is taking less energy and producing more ATP. Thus, I hope that this will help you in understanding the process of photosynthesis. Thank you.